Here we are looking at the fabrication of PRISM. Each component is brought into solution to create the sponge. First, gelatin is added. This provides the mechanically strong backbone of the sponge. Collagen 1 is added next. This extracellular matrix protein provides elasticity to the sponge while promoting a comfortable environment for the muscle-resident stem cell, the satellite cell. Laminin 111, another extracellular matrix protein, directs satellite cells to the sponge. By bringing these regenerative cells to the sponge, we hope to promote myogenesis at the defect area. FK506 is an immunosuppressant used in organ transplantation. We add it to our sponges to slow down the immune response to avoid the damage that comes from activating immune cells. Finally, we add EDC to the solution to act as a cross-linker to strengthen the sponge and prevent rapid degradation. We follow a slow cooling protocol to freeze the sponge without damaging the structure. After freezing, we lyophilize the sponge to dehydrate it, while leaving behind a macroporous structure large enough to allow cells to migrate into it while being compliant enough to withstand the harsh in vivo environment. PRISM can be made in any shape, mold, and can be cut to fit. Here we are creating cylindrical biopsy punches out of the small sponge we created. These samples are going to be implanted in a rat model of VML after an ethanol sterilization process. We create a defect in the tibialis anterior in the rat hind limb. A defect of this size is unable to be recovered without any assistance. We add the PRISM to the defect, suture the skin back together, and let the animal naturally recover over 2 weeks. After 2 weeks, we found that the animals left untreated showed fibrotic scar tissue deposition signified by the red bands of collagen. The animals treated with PRISM show muscle regeneration in the defect site at 2 weeks. This can be seen by the small diameter green myosin positive fibers growing in the defect.